Hello, everybody. My name is Christophe D'Enfert. I'm the Scientific Director of Institut Pasteur. I'm very pleased to welcome the more than 700 participants to this webinar on COVID-19. So we've decided to have this webinar in the frame of the MOOC entitled Emerging and Reemerging Viruses that was relaunch in a self-paced mode from May 31st to September 30. This is a MOOC that presents different viruses involved in viral emergence and describes the molecular mechanisms and conditions responsible for this emergence. So this MOOC was already broadcasted, but it was for this edition incremented by 5 sessions on COVID-19. And today, you will have the opportunity to ask questions to the speakers in this 5 specific COVID-19 session. So this session talked about the data on the virology, epidemiology, and vaccinology of COVID-19. Latest discoveries about structural determinants of SARS-CoV-2 for developing therapeutic monoclonal antibodies, impact of SARS-CoV-2 infection on the central nervous system, etc. So the MOOC was attended by about 3,000 participants from 108 countries, and I think it's a very significant achievement and this demonstration that the MOOCs implemented by Institut Pasteur have a very large audience. And today, before we go into the questions, I'd like to introduce our first speaker. Start by 𝐃𝐢𝐬𝐬𝐢𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬 who leads our vaccinology and immunotherapy initiative at Institut Pasteur. She's a vaccine specialist. She's worked in industry before joining Institut Pasteur. Arnaud Fontanet is an epidemiologist. He's the head of the Epidemiology of Emerging Disease Unit at Institut Pasteur. He's a member of the scientific council that advised the French government on how to handle the COVID-19 epidemics. Olivier Schwartz is a molecular virologist. He's the head of the virus and immunity unit at Institut Pasteur. Félix Rey is a structural virologist and is the head of the structural virology unit. And finally, 𝐃𝐢𝐬𝐢𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬 pathology, and he's contributed to the development of COVID-19 animal models at Institut Pasteur, and how we can apply these models to study vaccines, as well as understanding the pathophysiology of COVID-19. So today we are going to answer a number of questions that have been sent to us by participants to this webinar, participants to the MOOC, but you also have the opportunity to ask questions through the question and answer field of the Zoom. So don't hesitate to ask questions. They will be moderated, transmitted to me, and I will ask them in addition to the questions that we have prepared. So I will immediately start by asking the first question to Arnaud Fontanet. So based on real life, what is the efficiency of vaccination? How long will vaccination last? Is it efficient against new variants, and especially the Delta variant? So what do we know about the efficiency of vaccination, Arnaud?
Thank you very much, Christophe, and really pleased to be with you today to discuss all these different aspects of COVID-19. So this first question is a complex one. I would say that, first of all, to make it short, these vaccines are very efficient. And try to remember that 1 year ago, we never hoped that we would have vaccines as efficient as the one we have today. And the first clinical trials, which were published in November 2020, showed amazing results with mRNA vaccines with 95% efficacy against symptomatic infection. And no one ever thought we would have such efficient vaccines available in such a short period of time. Now the situation has become a bit more complex. It has to do with the fact that, as you know, we have several vaccines, which not all have the same efficiency. We also have to deal with different variants, and some variants may respond more or less well to the different vaccines. And now we also have to introduce a notion of time since vaccination, because with the Delta variant, the latest variant, which has now completely replaced other variants all over the world, it appears that the efficacy of the vaccine may change with time. To focus on the Delta variant, which is the most relevant one because it is now almost everywhere. What we can say nowadays is that the vaccine has remained extremely efficient when it comes to hospitalization. And regardless of for how long you have been vaccinated, we have now data with people that have been vaccinated 6 months ago. The vaccine remains very effective against hospitalization, which is the most important thing. It's against severe forms of disease, and we have a 90% vaccine efficacy up to 6 months after the second dose against hospitalization. The new findings were regarding the efficacy against infection itself, where we realized that with time passing since the second dose of vaccine, the efficacy against infection has decreased. It's to give a general estimate, I would say that eventually 4 months after the second dose of an mRNA vaccine, you are still 70% reduction of the risk of infection, and it may go down to 50, 60% after 6 months. And there are some new data which are going to be published in the Lancet this week with the Pfizer vaccine, to give an example. So it's not as good as it used to be. It means that if you are vaccinated, you are still at risk of being infected if you are away from your second dose. However, it is important to remember that if you are vaccinated and infected, you are less likely to transmit the virus to others compared to if you are infected without vaccination. So not only you decrease your risk of infection, but you decrease risk of transmitting to others. And again, as I was saying earlier, your protection against hospitalizations remain very good. So I'm not going to go into more details because we could try to discuss about the different type of vaccines available or whatever. I'm sure there are many questions, but that was just the first introduction I wanted to give you on this complex topic.
Thank you, Arnaud. Olivier, do you want to add something from a more experimental standpoint?
Yes, so the important question regarding the real world efficacy of the vaccines, of course, is to understand how do the vaccines work, and that's why we often speak about correlates of protection. So meaning, what is the biological parameter that we can measure and that will correspond to a protection against virus acquisition or against symptomatic infection. And so far, the best correlate of protection, if not the unique, I must say this is the unique, is the level of antibodies or neutralizing antibodies against the different variants. And there are multiple reports now in the literature describing a kind of threshold anti-RBD antibodies, anti-spike antibodies, or neutralizing antibodies. So now it becomes more and more simple or simpler to have an idea just by taking a drop of blood of the potential efficacy of the vaccine. And that's precisely what we are currently doing in our labs, measuring over time the evolution of the neutralizing antibody response, for instance. And this is also true in animal models, by the way, in mice or in primates. The best correlate of protection is the levels of antibodies. I can discuss later, if needed, the role of other arms of the immune system, including cellular immunity, innate immunity, which are also very important. But what is important, again, is the levels of antibodies.
Thank you. So there was a specific question about efficiency in immunocompromised patients. Chris, do you want to say something about that?
Yeah, hello everyone. So just a little bit to add on this topic. So there are special trials conducted also in immunocompromised, in let's say dialysis patient, cancer treatment patients. And what is typically recommended also from other vaccines is that they get one shot more than the general population. So also for the COVID vaccine, typically 3 shots are recommended, and efficacy has then seen to be about 65%. So it's not as high as is reached in the healthy adult population, but 65% is also not bad. So this is followed up more. There might be vaccines developed that target those groups even specifically, but initial efficacy even of the current vaccines looks decent.
Thank you. So we just received a question through the chat, and I'm going to read it. So how can we quantify the risk of being infected for vaccinated people versus non-vaccinated today? Is it possible to have a quantification of this risk? Olivier or Arnaud?
Again, I think that the levels of antibodies is very important. I just read this morning that there is an article in medRxiv by Andrew Pollard and colleagues from Oxford, and they defined a threshold of antibody levels. It's not an absolute protection, but if you have there is a kind of unit which is called the BAU, binding antibody units, that can be applied in all labs thanks to the WHO, and it's not an absolute protection, but if you have a certain level of antibodies, it is estimated that you have 80% of protection against symptomatic forms of the disease. And that's something that is very important to measure over time, both at the population level or in individuals at risk, I would say.
Thank you. Arnaud, you wanted to say something? No, okay. So we are going to move from efficiency to safety of the vaccines, and Chris, there is a question I think you could answer, which is how to be sure that there will not be long-term consequences of mRNA vaccines.
Well, I mean, maybe first to give a general introduction more about vaccine safety. So considering that we have more than 5.5 billion doses administered of the COVID vaccine so far, we have heard very little about side effects. I mean, of course, once you start immunizing hundreds of millions of people, then rare side effects that can occur become visible. So for example, the thrombopenia problem with the AstraZeneca vaccine, or the myocarditis with the RNA vaccines. But in both cases, you look at about 20 cases per 1 million population or vaccinated people. And at least the myocarditis is more frequent after COVID infection than it is after COVID vaccination. So just to say, vaccines are really carefully studied. I mean, in the phase 3 trials, there were already 30,000 to 65,000 people with a very careful follow-up the whole time. And we also have to take into consideration that neither the RNA vaccines nor the adenoviral vector vaccines were completely new. So the adenovirals have been used for decades. Basically, also the first mRNA or RNAs came 20 years ago for cancer treatments or vaccination. And the first infectious vaccines based on RNA have been gone into phase 1 clinical trials more than 10 years ago. So while these are maybe only thousands of people and not tens of thousands and millions as we have now, there have been no long-term effect being observed of those. So then the specific question was about the line 1 possible problem with the mRNA. Maybe I give to Olivier and then maybe come back if there's something to add.
Yes,
yeah, Olivier.
There was an article which was quite controversial, by the way, which described in a cell culture system, so in vitro system, that if you can overexpress certain cellular enzymes, then you can have little bits of RNA which are reverse transcribed, meaning that they go from RNA to DNA, and then they could eventually integrate. But other studies recently published in Cell Reports, for instance, clearly demonstrated that it was a kind of artifact observed in a special cell culture system. So my feeling is that there is no risk of RNA integration from RNA-based vaccine. Each cell produces tons of RNA every day, every hour, and there is no major integration of those normal RNA within the nucleus. So the vaccine should be considered as a normal RNA, and there is no risk of integration.
Okay. Do you want to add something, Chris?
Yeah, maybe just briefly. I mean, Olivier sort of jumped in. So in theory, the RNA goes into the cell, but as most of you will know, it doesn't go into the nucleus. So it's only translated into the protein. And once the protein is expressed, sometimes then the whole cell actually is destroyed at the end if it was too much, or at least they break down the RNA. So the chance event Olivier described, or not the chance event he described, but the one that was published in this controversial paper and doesn't look really real, would be really an extreme situation. But that's why I mentioned that also RNA vaccines have been used already for more than 10 years for infectious diseases, and so far no long-term consequence has been found from any of the other trials.
Thank you. So we now come to the question of emergence of new variants, and we have this question, which I read: is it true that vaccination will select variants which evade immune protection? So Olivier, maybe you're best placed to explain that.
So the variants emerge in multiple countries through through different waves, and they emerge, the main one, alpha, beta, gamma, delta, before the occurrence of massive vaccination. So that's not the vaccination which by itself created the variant. It's more the huge circulation of the virus in millions of individuals, which allowed for the selection of the variant. What can I add? The virus is the coronavirus is mutating very slowly. It is estimated that there is about one mutation every 10 to minus 6 nucleotides, meaning that every replication cycle, there are very few mutations occurring, because there is a protein which is called NSP14, which is a proofreading exonuclease, which corrects the errors which are made within the viral genome. Nonetheless, there are some mutations which appear when you have millions and millions of viral replication cycle. And that's the main origin of the generation of variants, which acquire mostly some mutations which facilitate replication, binding to the receptor, entry into the cells through increased fusion, and also escape to some antibodies.
Okay, thank you. So I have questions for Chris. So what about the design of Delta-specific vaccines, but also the design of broad-spectrum viruses that could target SARS-CoV-2 as well as other coronaviruses?
So for the Delta, the answer is quite quick. There are already 2 vaccine candidates currently in clinical trials. One is an mRNA vaccine from BioNTech Pfizer. They try the classical, the alpha, delta, and also a mixed formulation. And the other vaccine is the adenovirus vaccine from Serum Institute of India. That's in trials for the beta and the delta and mixed. So clearly more groups are working on it, but these are the first to enter clinical trials. Then for other coronaviruses or beta coronaviruses, I'm not quite sure how far the development is. I haven't yet seen any trials, but for example, CEPI, the Coalition for Epidemic Preparedness Innovations, has launched a call to support programs developing cross-reactive vaccines.
Okay, and another question. So about RNA vaccines clearly change the field. Do you think that RNA vaccine will replace any other type of vaccines?
Not any other type. So for example, many bacteria have a polysaccharide surface, and right now we are not equipped with RNA to make the machinery to build those antigens then in vivo. So clearly carbohydrate or polysaccharide capsular vaccines will remain for a while as there are. For other aspects, I guess we have to see also the longevity of the antibody response. Right now it looks at least the antibody titers go down quickly. Is it the COVID antigen or is it the technology? So if we think about new childhood vaccines in infancy that ideally we only get once, would RNA be the technology or not? But I mean, only time can tell. But clearly the bacteria or the polysaccharides will pose for a long time quite a challenge for RNA. So no.
Okay, thank you. So I have two last questions on the vaccine aspect. So the first one is about whether there is a correlation between the number of injections and the level of antibodies, and this refers to the use of a third dose. So Olivier, maybe you can answer very briefly that question.
Yeah, the use, it is clearly demonstrated now that when you inject a third dose, you strongly increase the levels of antibodies by 10 to 100-fold. And this booster effect is also seen when previously infected individuals are vaccinated. So that's clearly a strategy to increase the durability of the protection mediated by the vaccines.
And the second question is for Chris. So you addressed the side effects at early stage of vaccination, but the question of very long-term complications, 10 to 15 years after vaccination, do they still stay true or not? Well, is there a risk at long term?
As of now, we don't see, because I mean, if it was really linked to integration, I mean, that would be so unlikely to happen that it's not expected. But that's why I highlighted again that the technologies are not novel. So they have been used for 10 years. Typically the subjects, if something, even once the trial is finished, subjects, if something strange would occur, they would go back to the clinical trial site. It would be captured by pharmacovigilance experts. And we have not seen anything come up long-term from adenoviral vectors or also from the mRNA. Of course, as I said, it wasn't hundreds of millions, but from my perception, it's unlikely to occur.
Okay, thank you. So we'll move now to questions about vaccination strategy at the population level. And the first question is very theoretical, and it's for Arnaud. What does influence R0, the reproduction number, and is R0 stable for a given variant?
Yeah, so R0 is what we call basic reproductive number. It's the number of secondary cases for each person who is infected in a population which would be totally susceptible to infection, and in the absence of any measures to control the circulation of the virus. So for the SARS-CoV-2, for instance, it was estimated at 3 with the original virus that circulated in 2020, moved to 4 with the Alpha variant, and now with the Delta, we believe it's somewhere between 5 and 6. So that means that with the Delta variant, for instance, if we were introducing the Delta variant in a population where nobody has ever been immunized through natural infection or vaccination against the Delta variant, and in the absence of any measures to control the circulation, each infected person would infect 6 other people. That's what the R0 means. And there is a relationship, and I think this is what this question refers to, between this R0 and the vaccine coverage or the population immunity you should reach before the virus stops to circulate in an epidemic mode. And in a population where people mix homogeneously, you have the same probability of meeting somebody as anybody else, then there is a very simple formula that tells you that the vaccine coverage that you need, for instance, to stop the epidemic circulation of the virus, which we call herd immunity, is given by the formula 1 minus 1/6. So sorry, 1 minus 1 over R0 to be more general. So at the beginning, when we thought that we would live with this virus of 2020, which had R0 of 3, 1 minus 1 over 3 was giving you 67%. And we were saying the virus will stop circulating in an epidemic mode when 67% or 2/3 of the population will be immunized. This has changed since, because now, unfortunately, we have viruses which have a higher R0. And also because we have realized, unfortunately, that with time passing, the protection against infection is no longer close to 100% as it used to be, but goes down to 70, 60% with time after 4-6 months since the second dose. So we no longer can stop the circulation of the virus, even among people that have been vaccinated. And it means we are, so we'll have to revisit a little bit this concept of herd immunity in a way of stopping the circulation of epidemic virus. I think what now is really important for us is more the issue of protecting against the severity, because this is what brings you to hospital and also what puts some stress on the health system. Now, just to finish on that question, there was an assumption about people mix homogeneously. You know, we all have the same probability of meeting other people, and we all know that it is not true. There are some people that have less contacts compared to others. If you take this consideration this fact into consideration, and there has been modeling on it, then indeed, for instance, when I was talking about a 67% herd immunity threshold with the first virus of 2020, with modeling taking into account the fact that people don't mix homogeneously, we could go down to 50%, you know, which was, it was not necessarily needing 2/3 of the population to be immunized, but half of the population before the virus stopped to circulate in epidemic mode. But as I said, this is old story. Now we have viruses with higher R0, and we have also vaccines which are no longer 100% protective against the infection. The other questions about the what influence R0, is it stable for a given variant? It's not exactly the same. As you understand, there are some biological properties of the virus which make it more or less transmissible, for instance, and that can be influenced by environmental factors. And we have some good evidence to believe that this virus, for instance, circulate better when the weather is cold. And that has been true for other respiratory viruses. And it does explain partially why the winter, for instance, is a season where SARS-CoV-2 circulates better. It also has to do with the fact that people tend to live in closed environment when it's cold outside. But we understand that there is something about the intrinsic property of transmissibility of the virus at different climate, and that probably plays a role. Currently, for instance, when you look at what's happening in Europe, we have nice weather and we see the circulation of the virus, which is going down. And just remember September 2020, we had exactly the same story. 1 week, cold weather, end of September in 2020, and the second wave was starting in all European countries. So these environmental factors do exist. The other element which can influence the R0 is the pattern of contacts in the population. If you have a population very dense and with a lot of contact between people and transmission goes through the respiratory route, for instance, for a given virus, the R0 would be higher compared to the one that you would have in a place where people live very isolated and have much less contact. So you see that it's something that can change depending on the context and some environmental factors.
So I have another question for you. I think our attendees would like to know your views about a worldwide vaccination strategy. So should we go for global vaccine coverage? Should that be a priority instead of having the third dose for rich countries in order to stop this pandemic?
Yeah, this is a very complex question. I mean, there is different issues which are behind this question, you know, about what should we do the third dose or give it to countries who really need it. There is a question of equity, of course. And if you are the director of WHO and your concern is the safety of the world and of all people living in the world, you would say we should have a more fair distribution of the existing dose of vaccine. And maybe the priority would be more to send the those which are available now to low-income countries to protect their vulnerable population before we give a third dose in industrialized countries which have already benefited from a full vaccination. And that's a very fair point. There is also another issue, which is that for the safety of the planet itself and all the people living on the planet, if we want to stop the emergence of variants, as Olivier was saying, we need to stop also the circulation of the virus, because this is what gives it opportunities for mutations and eventually emergence of new variants. So there is also a global interest in vaccinating the entire planet for protecting the world against emergence of new variants. My last point would be to say that I myself, of course, would really advocate for having the entire planet vaccinated. But it's not going just to be a question of dose. It's going to be a huge enterprise to get the planet vaccinated. And I'm a little bit afraid of this short summary that we do in saying, oh, let's take the third dose of industrialized countries to give it to low-income countries so that we help them protecting their vulnerable population. I fully agree that this is a very fair statement. But if we are serious about vaccinating the planet, then it's not only a question of dose. It's a question of bringing those doses, organizing the logistics which goes to it, the delivery. There could even be questions of perception of vaccines in some countries where people feel like, why should we be immunized? Is it really necessary for us or whatever? All that will require a lot of effort on the long run. It's not something that would be do just like snapping fingers. And there have been studies by the International Monetary Fund which show that those are bulk figures, and take it as it is, but it would cost $50 billion to immunize the planet, and it would save $9,000 billion. So the ratio, is clearly a cost-effective operation, but it's $50 billion to start with. And it's not only money, it's not only dose, it's all the logistics and all the information that we need to accompany this effort. So I just don't do a shortcut in saying, oh, let's give the dose and this will make it. I think it will require a huge effort. It was done in 1979 with the eradication of smallpox. But remember, we had a highly effective vaccine, one dose, the symptomatic disease, so that it was easy to identify people that were infected and therefore where vaccines had not been delivered, and lifelong immunity. So we are not there yet.
Anyone wants to add something on that? No, so if not, I suggest that we move to the next topic, which is treatment of COVID-19. And so there is a quite specific question for Felix about whether other therapeutics could be developed that would block the RBD conformation change and prevent virus entry. And would that be safer than a vaccine? Your microphone is off, Felix. Yes.
Okay, is it better now?
Yeah.
Sorry, thank you for the question. So yeah, so definitely the RBD, as you know, the RBD, the virus needs to interact with the receptor in order to enter cells, and the RBD, the surface that interacts with the receptor is not exposed in a closed pack, so it has to open, and there is a dynamic there. So molecules that can block the RBD so that it does not expose could be good antiviral targets, and there are people trying to identify molecules that add that, or any other way to interfere with interactions with receptor. But what you need to bear in mind is that a single inhibitor, the virus, especially for RNA viruses, even if there is this NSP14 protein that controls the efficiency, they will still develop resistance to this approach. So what needs to be done is something similar to what was shown to work with other viruses, like for HIV or for the hepatitis C viruses, to target simultaneously different steps on the viral cycle. For instance, target the RBD, but at the same time target the protease. The viral protease needs to cleave at a number of sites on the polyprotein precursors that are generated, and this is crucial for the virus to replicate. And in addition, there are other sites like other molecules targeting the polymerase or some other enzymes that are strictly necessary. So these approaches are still, it's very recent for HIV. There was like more than 20 years of work in order to come up with molecules that work like that. For HCV, it is possible to cure now, but also it was discovered in 1989, and this cure came in 2014 only. So you can see the time that it will take. So now we're only 1.5 years or 2 years after the epidemics, and I'm sure that this type of treatments will become available, but we need to give it more time.
So your view for the future of viruses cure is really a combination of vaccination and then therapy, but combination therapy.
Exactly. So the vaccination, as long as we cannot do much, we don't know if this will become something like the flu, in which you will have to have a different vaccine every year. So coronaviruses are supposed to not be that variable as influenza, but we are learning as we go that this virus can find mutations as well. So in the long run, I think it is not as difficult a case as HIV, in which a cure it doesn't get integrated into the genome of the cells of the individual. So to eradicate it, it's more like an hepatitis C virus, but there is different, because the hepatitis C virus is a chronic virus that once you infect it, it will stay in your organism all the time. And so then to cure, you have this step. Here you have recurrent infections every season. And so I think there is a lot to learn here.
So actually there is a question that is related to that, and it's probably a question for Olivier. Does SARS-CoV-2 mutate more than any other coronavirus?
No, I don't think so. I already discussed earlier the mutation rate. We know that for coronaviruses in general, it's about 3 to 10 to -6. By the way, it's tenfold less than for HIV, which mutates much more efficiently. But as far as I know, it's the same for all coronaviruses.
And what do you know about the mu variant?
And so just to finish, to add,
oh, sorry,
for the other, it's not the mutation rate which increases the diversity for SARS-CoV-2, it's the number of people who are infected. So it's the large, huge circulation worldwide which generates these mutations. And what about the mu variant? As far as I know, it's not that problematic. It was originally described in South America, in Colombia. In last July, it represented about 0.6% of the sequences which are deposited on 𝐃𝐢𝐬𝐢𝐬𝐢𝐬𝐢, the database, and now it's less than 0.1%. So it doesn't seem to resist the invasion by the Delta virus, but we will see in the future. So far, it doesn't seem to be a large problem. And that's true. It is close to the beta variant in terms of sequence. And it's true that there is a kind of trade-off between the different variants and the mutations. If you introduce too many mutations which may allow antibody escape, then you will lose viral fitness and the virus won't be able to spread efficiently between individuals. And with Delta, there is a kind of equilibrium between antibody escape and viral replication, which is at the origin of the spreading of this variant.
So we have a question in the chat that I think is interesting, and Felix, you probably have an answer to this one. Is it possible to anticipate single mutations which enhance the affinity of the spike to its receptor and then use that as a predictor on how variants that emerge in the population will behave?
Yeah, so definitely there were some of the mutations are positioned for 17 or 501 were predicted beforehand by, it was a group in Israel at the Weizmann, for instance. They have a paper showing that which mutations would enhance the interaction with ACE2, with human ACE2, and they actually turn out to happen. For instance, 501 or this 484. So that's definitely true. The number of predictions, I mean, you cannot predict which one will happen, but you can predict that if a mutation for which a given residue at that site would increase their affinity, and that's what.
Thank you. And another question probably for Chris. So is anybody trying to do vaccines against other antigen than spike?
Yeah, it has started. So initially it was spike because from SARS-1 and MERS, basically it was clear that the spike would be a good antigen. And from preclinical models, particularly with the SARS-1 vaccines, there was this risk of disease enhancement, or it was observed when the N was in the vaccine. So that's why it was excluded from the early SARS-CoV-2 vaccine developments. But since we have not really seen any disease enhancement with SARS-CoV-2, people are now starting to include also other antigens for antibodies. And then, of course, there's also development towards T-cell vaccines, and then epitopes come from all conserved proteins, so not only the spike. So yes, more antigens included.
Okay, thank you. So there is a question that makes often newspaper headlines. Was so how did this virus emerge, and how can we be sure that it was not an accidental leak from a laboratory? So maybe Olivier, you can address that question.
I can start. This is a very complex question. What is clear is that there is an animal reservoir for the virus, for coronavirus, as for other viruses. And in the case of coronaviruses, the bat is clearly the viral reservoir. You may know that there are about 1,500 different bat species worldwide, and there are hundreds of coronaviruses which have been isolated from these different bats in different regions of the world, and some of them are very close to the SARS-CoV-2, and there are continuously new coronavirus isolated from bats, and they are very close to the pandemic virus. So for me, it's clear that the virus comes from bats, and the question is how does the virus move from bats to humans? And in most cases, and this is the most plausible hypothesis, it's zoonose, meaning that somewhere in the virus jumped from the bat either to another animal, which is called an intermediate host, or directly to human beings. And it is clear that there is no, the fake news saying that the virus has been artificially created. There is no evidence. And when you look carefully at the sequences, you can see that there is no obvious evidence of manipulation. And the question remains open, and there are numerous articles and debates, and it's important to keep a scientific approach, look at the sequence, and how does the virus jump from bats to humans. So it could be that within a market, this was one suggestion, but in other places as well, the virus might have jumped, and it is also plausible, but it is not the most the most likely hypothesis is that within a lab in which people were studying bat coronaviruses, someone could get infected accidentally. This remains an open possibility, which is much more unlikely than the other one.
Thank you. Anyone want to add something on that? Arnaud?
Just
briefly,
to remind, just to remind the history of the SARS-CoV-1, which emerged in Southeast China in 2003 or November 2002, where you had a bat as a reservoir and the civet cat as the intermediate host sold in markets, and then that's how it jumped to humans. Currently, the only evidence we have of the presence of SARS-CoV-2 in that time of mid-December, where it may have emerged in human population to the point that it would become epidemic, comes from the market in Wuhan, and the one that was studied, where they did find in what they call environmental samples, positive PCR from a series of specimens they have taken in this market, and it's not in animals, and they found them. It's not really clearly described whether it's used water or in the places, wooden stalls, you know, that were used at the place where the animals were sold. But they did find and reported that there were a number of specimens positive for SARS-CoV-2, so that's the only evidence that currently exists. And it is known also, and there was a paper published in Scientific Reports describing that in that market, very large number of animals, including those animals that we believe could be the intermediate host. I mentioned the civet cat, but there is also the ferret badger, there could be the mink, there could be all those sort of animals like this. And these animals were sold in that market and could have been the intermediate host bringing it. Again, we don't have any evidence for this, and we have to keep an open mind and try to collect as much as objective information as possible on that.
So now there is a question on you know, the epidemiological status in Africa. And so is there any explanation to the fact that COVID-19 in sub-Saharan Africa is relatively limited?
Well, in fact, you know, what we have learned using serological studies, which look at antibodies against SARS-CoV-2 in different surveys performed in African countries, is that the virus has circulated a lot in Africa. And there was recently a paper in JAMA, for instance, where they found that populations in different cities of Kenya had up to 60% of antibodies against SARS-CoV-2. And there had been already a paper in Science, again from Kenya 1 year ago, where it was about 15%, 1.5, which showed that the virus is circulating there. There was a paper also from Kinshasa recently in CID, again showing prevalence up to 30% in the adult population. So the virus is circulating there, but most likely because of a very young population, the burden of disease associated with the circulation of the virus is very different from what we live in industrialized countries. Remember that the ratio of the risk of dying is 20 times higher if you are above 60 years of age compared to less than 60 years of age. And in countries, so I don't know specifically for different sub-Saharan African countries, but for instance, India, only 10% of the population is older than 60 years of age. So you can really have a very different impact of the circulation of the virus when you have such difference in age distribution and a much lower proportion of people above 60 years of age, like in many of the sub-Saharan African countries.
And there is another question about the real impact of COVID-19 in children. Can you address that?
Depends a bit what we mean in impact, but just to summarize briefly, what we know is that COVID-19 is not causing severe disease in children, or very rarely. The risk of hospitalization in France, for instance, was estimated at 1 per 1,000 infections. So it's much lower compared to what we have in adults when it comes to children. There are some particular complication, inflammatory syndrome in children, but which remain also extremely rare. And there were, we count that in few hundreds of cases for France, for instance, during the first and second wave. The message is really to say that children do benign forms of infection, and it's a very good thing. I mean, I can't imagine what this epidemic would have been in terms of our feeling about it if children had been hardly hit by this epidemic. Now, when it comes to susceptibility to infection, what we can say is that for the children who are less than 10 years of age, they are probably less susceptible to infection compared to adolescents or adults, maybe 50% decrease in risk of infection. They are also probably less contagious, but it may have a lot to do with the fact that they do asymptomatic forms of disease, and people who are asymptomatic transmit less. Adolescents, I would say, are probably as susceptible to infection and as contagious as adults. But again, there is this incredible gradient of severity of disease with age, which means that adolescents are still at a stage where the risk of developing complications is low. And now there is a pending question on long COVID, which is something that we are going to address soon, I guess, in this webinar. But in children, it seems that the risk of long COVID is lower compared to adults.
So we've lost Arnaud. I hope he will be back for the conclusion. So we lost you a little bit, Arnaud. But I'm going to ask a question to 𝐵𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬 can we be sure that the virus is not latent? And so this could contribute actually to long COVID.
Yeah. Hello, everyone. Thank you for the question. But the common long persistent symptoms in COVID-19 patients is usually dyspnea, chest pain, and fatigue, which are related for at least 30% of these patients. But also the report of persistent anosmia, the loss of smell and taste, and even other more psychosocial or cognitive disturbance, and even anxiety or depression. In some cases that we studied, people with long-lasting anosmia with a negative PCR test on nasal swabs, we could detect actively a virus and inflammation, SARS-CoV-2 and inflammation in the olfactive mucosa of these people. So one possibility is that persistent virus and inflammation, that could be the one of possibility of these persistent symptoms. And for latency, it's difficult to say because we don't have actually the whole elements to answer of this question, because a virus should be latent, it must be also persistent, but also to be reversible. And we don't know if patients that was positive for SARS-CoV-2 at the beginning and then present a PCR, a negative test, we don't know if the test was appropriate. As I said previously, the nasal swab, a negative nasal swab test is not true that the virus was cleared in the whole organism. The virus can persist in the olfactory mucosa. We can see reports that the virus persists in the gut and also in the peripheral nervous system. So it's still an open question, but the persistency, the long COVID case, is probably related to viral persistence and also to inflammation persistence in this case.
Thank you. Does anyone want to add something on long COVID? So if not, I move to one question again to Arnaud about whether modelization has proven useful in this pandemic, and what are the limitations of mathematical modelization in a pandemic?
So in short, modeling has been extremely useful in this pandemic, but it has not always been well understood. I mean, what it means. And I think the very important part to understand is that the modeling allows you to simulate different scenarios and tell you, if you do this, this is the number of cases you could expect. And if you do that, this is the number of cases you could expect. But it has never said, this is what is going to happen. There was always this if, and people had tend to forget this if. So some modeling, which were showing different scenarios, will eventually be portrayed by the media who like to bring some sort of sensational news, taking the worst possible case scenario and saying, this is what the model is telling us will happen. And that's not at all what was in the model. The model was saying, if you don't do anything, for instance, to control the circulation of virus, you will have that many cases at that date. But of course, if you do something, you will have much lower number of cases. And it's just to help the policymaker, you know, in choosing among different options, which one is the most feasible for the policymaker, and at the same time would have an impact on the epidemic. Once you have understood this, models have been incredibly helpful in this epidemic. And as a member, for instance, of the French Scientific Council, I can say that we would have been in a very difficult position if we didn't have the support of these models in helping us advising the government. But of course, many people have said all those models were totally wrong, and they were saying such and such, it never happened. I would say well, we are all happy it never happened, just because people took measures based on the models and chose the measures that would best control the circulation of the virus. So that's where, but again, models have been extremely useful.
Thank you. So I'd like to finish by asking you a last question about preparedness and future. So what are the lessons we've learned from this pandemic in terms of preparedness at the nationwide level, but also worldwide level? So what could we improve? And will we eradicate this virus? How can we predict the emergence of new zoonoses and be in a better position to fight against this zoonosis? So I don't know if any one of you want to address this question and give your opinion individually on these matters.
Well, maybe I will continue and just, sorry, Chris, but
go ahead.
and try to be, I mean, the question, of course, I mean, cannot be answered even in a few minutes. It's too complex. I think what I think is really important is that this pandemic unfortunately showed us that these things could happen. And I hope that the lessons will be taken so that we are better prepared next time, and that we will be heard about recommendations in making us better prepared, because I think it's a normal psychological phenomenon, but we tend to forget immediately after a crisis. And but this time the crisis has been so severe that I believe it will have lasting effects, and people will be more ready to listen to the recommendations made. I would not be able to list them; there would be too many. But at least we understood that this sort of pandemic are a real risk and can happen. And I would say regarding the future of this epidemic, it's not over yet. We don't have the end of the story. I think what we see happening now is more a move towards what we call endemicity, with coronavirus that will continue to circulate, with probably seasonal surges during the winter. But hopefully, with the help of the immunization of the vulnerable population and a few things that we have learned in order to control the circulation of virus at peak, which we were not using before, but now we would consider, I think, more likely, we will be able to not to suffer to the same extent as we have suffered since the beginning of this epidemic, and avoid the major lockdowns, curfews, and measures that had to be taken. And it will be more something like what we see, for instance, to take an analogy with the flu, where in the fall you immunize people and the vulnerable ones, and you still have a bit of tension during the winter, but at bearable level as compared to what we have seen before.
Chris, you wanted to add something?
Yeah, maybe not to add, because it's not really related to this specific pandemic, but let's say from the vaccine perspective, just to give a bit of context. So when SARS-CoV-1 came in 2003, it took several years actually to start the first phase 1 trial before, and at that time, the outbreak was already over. With MERS, it was between 1 or 2 years, I don't quite remember. And now we had the vaccine licensed in 11 months. So, and even that now, some people consider it could be even faster. So I think on the vaccine development perspective, we have learned a lot, let's say, where particularly also clinical trial design, how the process can really be speed up. Clearly, a next pandemic with the virus that's amenable to RNA technology, I think that will be the fastest to do, and vaccine production capacity is now there. But also for other technologies, the ways how to be fast without cutting corners, because that's important for the safety aspect. I think we have learned a lot. So I think next time it will be even faster. And there are a lot of programs trying to predict also maybe which diseases are more likely to come up. So there will be more and more emphasis of sort of putting vaccines already on the shelf for the pathogens that are considered the most likely.
Felix, you wanted to say something?
Yeah, so
very brief, because we have 1 minute left.
Yes, so for what Christophe said, is because there was a 2003 SARS outbreak and then the MERS, that we knew exactly what we needed to put and what mutations to make a stable variant, and we knew a lot of things that we wouldn't have And so what we have learned that for other pathogens that may emerge for which we don't know anything, which we should start studying now because they might emerge later. And the other thing is that this virus may, in the end, become like another seasonal coronaviruses. When the immunity, the general population becomes immunized, it's probably to stay. And if you think all of the previous seasonal coronaviruses have zoonotic origins. And when they first entered the human populations, they probably were much more virulent. And with time and people immunizing have calmed down. So we don't know what really happened. And we might be witnessing the entrance of one new virus that will stay in the human population.
Thank you. Olivier, you wanted to say something? 𝐃𝐢𝐬𝐢𝐬𝐢𝐬𝐬𝐢𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬 answering these many questions that were sent by our 700 attendees. I'd like to thank everybody for participating in this webinar. This webinar has been recorded and it will soon be available on the Institut Pasteur Education YouTube channel. For those who have participated in the MOOC, you know probably that no certificate was issued for attendance to the MOOC, but you can register for the next certified edition of the MOOC, which will be scheduled early 2022. And you can also, if you have validated 5 MOOCs of Institut Pasteur within a 2-year period, obtain the Institut Pasteur online diploma in infectious disease. And we all thank you for participating in this webinar. I think it was very a fruitful discussion. I thank everybody for questions and all our speakers for their answers and contribution to the webinar. Thank you very much and good luck in the pandemic.